pathways as well. Right, so I think that's it. I'm going to hand over to Jamie now to introduce us to Matthias and Shareen. Over to you, Jamie.
Hello, everyone. Welcome. It's a very windy but sunny Friday where I am in the world, so I hope it's a very nice day where you are too. So today we're going to be finding out all about islet transplantation, stem cell-derived beta cell therapies, and much more information on the Diabetes Grand Challenge. And as you might have guessed by me not wearing a lab coat and a hoodie instead, I'm not the scientist. I am just a professional speaker of words. So I'm going to be asking questions to two very esteemed professionals, scientists, who are joining me today on this webinar. So we've got Professor Shareen Forbes, who is a professor of diabetic medicine at the University of Edinburgh, and Professor Matthias Hebrok, who is a leading scientist in the field of stem cell and diabetes research based at the Technical University of Munich. So very, very interesting work that these two people have been doing today, which we're going to be finding out about as well. And a couple of the words that I said there might have raised a few question marks in your brains, as it did mine when I got to reading some of the sort of information that was given to me beforehand. But don't worry, we're going to keep it all very understandable for you today. You don't need to ask ChatGPT what we're talking about. If I don't understand it, you're not going to understand it. But if I do, you're likely to. So we're going to get through this together in a way that hopefully you'll be able to understand. But if you do have any questions or thoughts or comments throughout today's webinar, pop them in the Q&A box. And then if you see one that's particularly interesting to you, or the same question that you were going to ask, vote it up, and those will get to the top of the screen and we will be able to ask those to the professors. So let's get going, shall we? We've got quite a lot to get through, so let's not waste any more time waffling on. So we're going to start with Professor Shareen here. Question for you to begin with. So for those who might not be familiar with this whole thing, can you explain a bit more about how islet transplantation works and when it first became available in the UK?
Yes, so many thanks, and thank you for inviting me onto this webinar. Really pleased to be here today. So islet transplantation is really a process whereby islets, which are the insulin producing cells of the pancreas, are isolated from a donor pancreas and actually transplanted into somebody with type 1 diabetes. And what happens is that a donor pancreas actually goes to the laboratory, islets are enzymatically taken out, and just a small, that small fraction of those insulin producing cells are actually infused into that person living with type 1 diabetes, into their liver, with the major vessel going into the major vessel going into the liver. And the whole procedure is actually done under radiology guidance rather than it being an open surgery, and can actually take place within 30 minutes. And what these islet insulin producing cells then do is obviously produce insulin in response to glucose levels. So they can increase the insulin secretion when the blood glucose is high, and they can shut off when the glucose levels are low. So it can really stabilize blood glucose control.
So the transplantation, I thought I was saying that word wrong then, but I absolutely wasn't. I'll be confident in my pronunciation of that. So that's been around for quite a while, hasn't it? So, you know, what have you seen from people who've had this procedure? You know, if their ability to control increased? Have you cured them completely?
Absolutely. So it's been around. The actual landmark publication of this was in the year 2000. And in that year, the Edmonton group actually showed that they could get 7 people off insulin injections completely. So it was a really landmark study which set in motion certainly our islet transplant program within the UK. And yes, we can absolutely get some people off insulin injections. We've seen that their blood glucose levels can stabilize, that they have far fewer hypos, and that, their levels of independence, not just insulin independence, but actually day-to-day living really improves. They feel much more autonomous. They can get their lives much more back on track, regain their driver's license, have better quality sleep, and just all those things that can contribute to improved quality of life can very much return.
Well, I'm sold. I think, when you talk about transplants, you're actually you'd be amputating my insulin pump from me, and I think I'd still get like sort of phantom annoyance from the insulin pump that isn't buzzing anymore under my pillow, which all sounds amazing. But are there any downsides to a treatment like this?
Absolutely. So the upsides are that obviously insulin independence can occur, but actually with human islet transplantation, that's normally in the UK, we get rates of like 30% insulin independence. And actually, most people require at least 2 islet transplants, first of all. The other major issue is, of course, that this is a treatment for adults only because anti-rejection drugs are required, and they can have quite serious effects in terms of exposing people to an increased risk of infection, increasing the risk of certain types of cancer, most notably skin cancer, and also those anti-rejection drugs can have an effect on kidney function. So there are downsides, and I think those need to be really recognised.
Yeah, so it comes with, at least adequate sort of serious considerations if this was to be rolled out on a larger scale. But thank you for that for now, Shereen. We're going to speak to Matthias now. So could you explain what stem cell derived beta cell therapies are? So we've heard a lot, and we continue to hear a lot about stem cells, Matthias, in the news, but it can feel a bit abstract. So can you break it down for me? What is a stem cell derived beta cell therapy?
Sure. First of all, thanks, Jamie, for hosting this and the grand challenge. It's great to be here. Yeah, when we talk about stem cells, there are essentially 2 different cell types we are talking about. And these are either what we call the embryonic stem cells that need to be isolated from blastocyst of a human unborn being, and so-called IPS, or induced pluripotent stem cells. Now, these embryonic stem cells have been used for a while now, and they were identified in 96 or 97. And people use these human embryonic stem cells to understand how you can form organs. And the reason for that is that these are the cells that, when we are embryos, essentially have the capacity to form every single cell in our body. So those cells, we call them toti, or complete, potent. They can turn into every cell that we have in our body, including, of course, the cell that we're interested in, the insulin-producing beta cell. Now, the problem already I mentioned is that you have to destroy life to get to the point that you isolate these cells. And therefore, approximately 10 years or so later, in the mid-2000s, there was evidence that we can take cells that we have in our adult bodies, for example, from the skin or from the blood, and by giving them a cocktail of just 4 or 5 different factors or genes, we were able, or the field was able, to take these cells and turn them into stem cell-like cells. They have the same capacity to also turn into every single cell that we have in our body, including the insulin-producing cells. And so the benefit of this is, if you either work with ES cells or these induced pluripotent iPSC cells, that now you don't have a problem of supply anymore. These cells can be kept in our laboratories in cell culture plates, and they can be kept for a very, very, very long time, and they maintain their ability to turn into the cells that we would like to have, the beta cells, the insulin-producing cells.
And Shareen mentioned a donor organ. So is this helping to address some of those sort of supply and demand issues, for want of a better phrase?
Absolutely. That is the idea behind it. Yes. With the cadaveric islet transplantation that we have, we depend, and Jean just said it, that often you have to have two donors. And unfortunately, that means that someone has to die to isolate the islets and then give them into a patient with diabetes. Now, for the stem cells, one of the biggest promises here is that you can take these cells, turn them into beta cells, and make as many beta cells as you would like to have. So therefore, yes, the supply and demand problem goes away because you can produce as many cells as you would need to treat a patient.
Cool. So it's you mentioned that there was a sort of big change at around, I forget the year that you mentioned, 2010 maybe, where we discovered we didn't have to use sort of embryonic cells. But in the past decade, because we are 25 years on from the turn of the millennium, what are the biggest sort of milestones that's happened in this area of research?
Yeah. The first thing I should say is that this field has been moving with lightning speed. To just set the stage here, as many in the audience might know, insulin, which is absolutely a miracle drug, has saved countless lives, was first used in 1922. Now, over the span of now a century, not just 25 years, we have moved from insulin from cows or from pigs to what we call humanized insulin. We now have fast-acting and long-acting insulin. But what we really want is an insulin that I inject in the body, and then it stays silent until it needs to be active when the glucose levels increase. We still don't have that. For stem cell therapy, again, these embryonic stem cells were identified towards the end of last century or millennium. And within less than 25 years, we've gotten from the idea of being able to take these cells into actually practice. They are already in clinical trials right now. They are being used in patients with type 1 diabetes, and they show some remarkable success. So what are the key milestones that happened? Well, if you think about this, let's say you are in London right now and you want to go to Scotland. In the beginning, you have to take these cells and you give them specific molecules to get them out of London towards the north, towards Scotland, and we couldn't even do that. For the first 5 years or so, these, what we called at that point, the embryonic stem cells, we were not able to even generate the first step on their way of going to Scotland. We would call this definitive endoderm. This definitive endoderm is a tissue that we have in the embryo that forms all of our tissues, starting more or less from the mouth, a little bit further down towards the rectum, and that includes the pancreas. But you had to understand what signals you have to give at the right time for the cells to move further north rather than going somewhere else. And that took a long period of time, and we and others in the field studied how the embryo makes that, identified the signals, the signaling pathways, the duration, the concentration, the combination of these factors to be able to guide them in the right way. We have more than 200, probably at this point, 250 or so different cell types in the body. So therefore, that the cell can give rise to any of those cell types also means that we have to be very precise in telling them exactly what path they have to take. So that was one of the biggest things that has happened over the last 20 years, to be able to replicate what happens in the embryo when our beta cells form, and to be able to take this and move this into differentiation that we can accomplish in the laboratory.
So what needs to be done now then to make the beta cells created from stem cells, sort of market ready?
Well, market ready is in the eye of the beholder. There are clinical trials. There are companies out there that are using them. They've been using them for years now. In the most recent meetings that we had, for example, at the American Diabetes Association, but also at a transplant meeting recently, there were updates. There are patients now out for more than a year that have been treated, and many of those are essentially normoglycemic. What that means is they don't have to take insulin anymore. The transplant, the stem cell-derived transplant, already regulates their glucose levels. So that is, again, it's a huge success. Having said this, these cells, when we produce them, they're probably not exactly like the cells that we have in our body. They're pretty good, but after transplantation, they still take some time to fully mature, to fully get and grow into their own, to have the full functionality. So what people are doing right now is they're trying to understand what is missing in the cocktails. How can we make these cells even better, probably better functioning, longer lasting, metabolically more stable? The second big issue is the one that Shareen already has told us about, is that the cells, even if they come from stem cells and they are being given to patients, the patient's immune system will recognize that these stem cell-derived islets or beta cells do not belong to them. They are foreign entities. And our immune system is very good, and that's why we're all alive, very good in identifying things or cells that do not belong to us and eliminate those. And so now there's a lot of effort in the field to think about how we can modulate these stem cell-derived cells so that they are not being recognized by the immune system, or only recognized a little bit, but not destroyed. And this is something that is going to be really critical to go forward.
Absolutely. Doing all this work to get them ready, functioning, and in there, then my own body goes and destroys them for you. That's not going to be very efficient, is it? But back to Shareen now. So we've got islet transplants happening already on the NHS, Shareen. So who meets the criteria? Who is available to get this currently?
That's a good question, because you've already talked about the supply and demand issue. And so at the moment, we reserve these treatments for people that have major problems with severe hypoglycemia, which is obviously low blood glucose requiring external assistance, as well as people who've got very unstable blood glucose control despite the best available treatments that they can manage. So not all our patients that we see in the islet transplant clinic are on insulin pumps or closed loop therapies, but if they can be on those, then this would certainly be a consideration. The other group of patients that this is open to are those people who who've got type 1 diabetes, who are being considered or who have had a kidney transplant, because these people are already on those anti-rejection therapies. And in fact, the bar, if you like, to transplant these people is a little bit lower because they are already on the anti-rejection drugs, and they may have issues with very unstable blood glucose control. And in fact, there's very good data now to suggest that these people really benefit from having an islet transplant, not only in terms of stabilizing their blood glucose levels, but also in terms of other complications, and it can be a life-saving treatment in them.
And what changes then in between an islet transplant and a beta cell therapy? What's the difference between the two procedures?
So, in fact, to myself, so islets are clusters of cells, so we use those terms interchangeably, really. So when we say sort of beta cell therapy, it's really to indicate that those beta cells are producing insulin, but they are obviously part of the larger islet cell cluster, if that makes sense. Obviously, I mean, Matthias may be able to kind of comment further with regards to the stem cell beta-derived therapy and the sorts of cells. So I'll let you answer that, Matthias.
Absolutely. So yes, we often talk about beta cells because we do know that in patients with type 1, it's the beta cell that is being destroyed. But the beta cells are sitting almost like in a little mini organ that we call the islet of Langerhans. And actually, they were first described by a human pathologist who looked at the pancreas and says, well, there's these little round things, little balls, they're swimming in the sea of the exocrine tissue, which is the rest of the pancreas. And he calls them islands or islets of Langerhans. But they are not just beta cells. Actually, they also consist of some other hormone-producing cells. And probably most notably, there is an alpha cell in there. That's otherwise we wouldn't have a beta cell. And the alpha cell produces a hormone that's called glucagon. And glucagon is the antagonist to insulin. It does the exact opposite. When insulin leads the cells in our body to take sugar from the blood and put it into the cells, glucagon makes the cells release sugar into the blood, so when we need energy. And yes, actually we had set out, and this is, I think, is true for most of us in our field, that we just wanted to generate a beta cell. But actually, because of biology, these so-called islet cells, hormone-producing cells, they all come from a common progenitor. And by us replicating the journey from London to Scotland, we were getting this progenitor, and that progenitor then led to the formation of the other cell types in the islets as well. So we definitely have alpha cells, and we also have delta cells. They are not as functional, at least in our hands, as the other cell types are, but they can become very functional upon transplantation.
Brilliant. Thank you for that. So, Shareen, then, what else needs to be done then to make beta cell therapies available in the NHS? What's the next step to bringing it into the healthcare system here?
I think, I mean, the islet transplantation is already available in the NHS. And has been for a number of years. So in 2001, the first research islet transplants were done in the NHS. And then it evolved into a kind of clinical program that is now free at the point of delivery. But the actual number of islet transplants that we do is still low. In the UK, we've done almost 350 islet transplants in the last 25 years in around 200 people. But in order for that to evolve, I think it's really important that we have a bigger supply of islets. And I think that will come from the stem cell community, really, and the clinical trials that are out there and happening now. We've had the first publication of the early phase clinical trials that the company Vertex have done, and that's caused a huge amount of excitement. And I think there needs to be more people enrolled in that trial still. We need to kind of see the safety of those therapies, because at the moment those therapies are going into the liver, which is quite difficult to survey in terms of safety. So it needs regular assessment in many different ways. And once we've had an adequate amount of safety data generated, then we hope that the NHS and other health programs can actually adopt these stem cell derived islet therapies. Many different companies, I should state as well, are making their own stem cell derived islets, and they're looking at many different sites to actually put these stem cell derived islets into, which might be easier to monitor in terms of safety.
And you mentioned a quick 30-minute procedure. So in my head, I'm sort of thinking about it like my sort of annual eye checkup. Go in, do the thing, wait for a bit, go back in, and then it's done. Once, considerable amount of effort has gone into getting this all up to speed, it could be a very efficient way to help people to manage and treat diabetes.
I think the procedure itself is one thing, but the actual giving the anti-rejection, giving the education to the patient as to how to manage their transplants, and the amount of monitoring that's actually required are the other downsides. And again, as Matthias has said, if we can get rid of the anti-rejection therapy, this sort of approach would make it much more speedy if the body didn't reject those cells. And not only that, I should state that if it was safer, it opens up the field to many more people who don't have problems just with hypoglycemia, but may have variable blood glucose control. And of course, if we can actually eliminate the anti-rejection drugs, that opens the field to delivering these therapies to children, which is really a very important point.
And then is there a patient that is not a particular candidate for this ever? Is there anything that would stop someone being able to have this in the future?
At the current time, the people that we cannot give it to are people who've got a history of cancer, because the anti-rejection drugs would probably promote the growth, or possibly promote the growth of that previous cancer. And people with who've got problems with kidney failure, who don't meet criteria for a kidney transplant immediately. So because again of the anti-rejection drugs, that again can cause problems. So a lot of it actually is related to the fact that the anti-rejection therapies can cause a huge amount of problems.
Well, I may chime in for a second here. The beauty of the stem cell derived islets is that you've asked me what were the milestones and breakthroughs we had over the last decades or so. There's another one that is not directly related to stem cells, but enables us to do stuff we couldn't do before. And this is this gene editing technology called CRISPR. People might have heard about this already. If you think about this, this is absolutely amazing. We have around 3 or so billion base pairs in our DNA in every single cell, and we have 10 to the 14th or something cells. It's unbelievable. In this case, the CRISPR can go into a cell and take out one of these 3 billion base pairs, and very specifically. So now we have the ability to modify the genetic code in a way to reduce the molecules that would activate the immune system. That essentially would lead to something I would call cloaking or immune cloaking. But at the same time, it has to be, of course, safe. If we were to succeed to generate such a cell that is being tolerated by the existing immune system and is safe and is fully functional, then I think, as Shareen just said, it would open up the number of patients and the classes of patients who could take this by a very significant margin. Now, the last thing I want to say is that, again, this has gone so unbelievably fast in 20 plus years to go from an idea to a concept to actually to clinical trials that are on top of this successful, at least from what the looks of it right now. But it's still, and I've spent too much time in America, but still, this is what I would call the Model T1. That Model T1 is the first model that was mass produced as a car from Ford more than 100 years ago. I can tell you one thing, my car that I'm driving right now does not look like the Model T from 100 or something years ago. And I think the same is going to be true here. We are just at the very beginning of this, and we have the ability to modify these cells. So I think they're going to get better over time.
Absolutely. It's exciting. And what's really exciting is the Type 1 Diabetes Grand Challenge, which you both are heavily involved in. So, Matthias, let's talk about what you've been doing to get the Type 1 Diabetes Grand Challenge underway, and your sort of most exciting things for the next 5 years of research.
I mean, it's the Grand Challenge is from Steve Morgan and Sally. And they have helped by providing a tremendous amount of funds to mostly, not exclusively, to the UK family of researchers that have an interest in working in diabetes and on all different areas. I'm involved on the SAB and the Scientific Advisory Board, and I'm helping or have helped to shape this a little bit in terms of the area of stem cell therapies. And I have been doing this actually now for 3 or so years, and it has been unbelievably rewarding because when we started out, again, it was a concept and we didn't know if this would work, but I think there's a fantastic team on the UK diabetes side and the Grand Challenge side that essentially enabled us to identify the researchers in this area, to bring them together and to ask them to come up with the best ideas how to move this thing forward. If you want to stay with the vision I gave you about the Model T, to move the Model T from 1910 or something to 1950 and then into 2000s. And we're seeing some applications. We had a meeting last year, and we're going to have another meeting towards the end of this year, where the first people came up and say, hey, this grant and this money has allowed me to really do some very innovative, in some cases risky research, but that needs to be done to leap forward and essentially hopefully get to quicker ways how we can treat patients.
And for everyone watching, if you did want to find out more about the Type 1 Diabetes Grand Challenge, there's a brilliant website with all the information there, type1diabetesgrandchallenge.org.uk. That's the number one in there. I'm sure you must have found your way onto that website at some point to know about this webinar. But Shareen, you are working on a beta cell therapy project for the Grand Challenge. Tell us about that.
Yes, thank you. And again, thanks to the generous funding by Steve and Sally Morgan, this has really been possible. So one of the things that happens with islet transplantation into the liver is that many of the islets don't survive past 24 hours. When the islets are infused into the liver, they don't have a blood supply, and they have to sit there for a while, and then vessels sprout and they form vessels eventually with the vasculature in the liver. And what my project is trying to do is try to accelerate that process of blood vessel formation so as to save more islets, if you like, so that they can be functional islets rather than dying islets. And it also addresses the fact that because so much immunosuppression has to be given, which can cause adverse safety effects in that patient, we're trying to actually deliver the anti-rejection drugs more locally to the liver so that the actual amount that's circulating in that patient can be lowered effectively. And so I've teamed up with a chemist who's making microparticles, and that's Dr. Lisa White, and she actually loads these microparticles up with different drugs, cytokines, to see what effect that has. And we're using animal models to test these effects, to test what actually promotes the greatest survival of these islets. And we're also looking at various different safety measures as well. So it's a really exciting project and very exciting time to be involved. And our hope is that then we will collaborate with people like Matthias and others who are in the stem cell world so that we can translate what we know with human islet transplantation and human islet engraftment, it's called, into the liver, that we can then apply it to stem cell islets, and that it's not just a treatment that might be applicable only to the liver, but in an era where we're seeing stem cell derived islets going into devices or other areas of the body, it might well be a treatment that can be applied to those other devices and other sites where they might go. So a lot of people are looking to put them under the skin, etc., where they can be retrieved.
I see. Interesting. So, that there, as you get later on in the stages, and you move out of the animal models and into potentially human clinical trial environments. What role can people living with type 1 diabetes or family members, loved ones affected by the condition, what can they do? The normal people, what can we do to get involved?
I think the main thing is to very much raise awareness about hypoglycemia, raise awareness about perhaps clinical trials that they might have been involved with or would like to be involved with. There's lots of panels that both type 1 diabetes, that both Diabetes UK and Breakthrough Type 1 Diabetes and other charities have, which actively want to recruit people with lived experience. And what's really important is that people with lived experience actually are collaborating with healthcare professionals, basic scientists, to try and put forward the best treatments for the future. But it's really important to raise that awareness, to raise awareness of what their therapies are. If people have had an islet transplant, they need to come forward. It really helps because the main thing that people look at is how does that patient feel with that therapy? How has it made a difference to their life? It's not just about the numbers, not just about, what HbA1c are we achieving? It's, how does it actually make you feel, and what has that meant to you and your family?
Fantastic. So don't forget, folks, do put the questions in the Q&A box. We'll get to those shortly. But Matthias, I just wanted to give you that same opportunity. As someone sitting on the scientific advisory board, what is the main thing that you think people in the community could do to help with efforts right now?
There's a number of different things. So I just came back from a Breakthrough Type 1 Diabetes meeting, the JRF, or formerly known as JRF, in the United States in Chicago. And I'm always, and actually took one of my trainees there, because I tell the people in the lab all the time that there's a reason why we're doing what we're doing. We, at the end, would like to use all of our scientific expertise and prowess and everything that we have to make the life of patients better. And so I think it is very important for patients to speak to us researchers, to essentially explain a little bit what they are going through. This is a disease that requires constant vigilance. I don't have to tell you that. You always have to be essentially informed about what you do and what you do with your body and what you put in. So essentially helping people like me to understand what kind of a difference our work could make, I think is inspiring to the people in the lab, because what we're doing as researchers is also very, very time consuming and it takes a long time. I also think that you probably should go out and talk to people in your community, including people who make decisions about where research and science should go, and to, in a way, almost nicely demand that essentially we are not stopping right now. We are getting really close to having a new technology, hopefully at some point widely available, that I really believe is going to make a difference in patients' lives. And we do need to have the support from the community to essentially continue our work.
Fantastic. Well, that's what webinars like this is all about, to help us understand what you're doing and how we can get involved. So there have been some questions coming in, and the floor's sort of open now to the two of you. So is the research, the data that we're collecting right now, is that helpful in producing treatments for other types of diabetes?
Absolutely. All sorts of biosamples that are collated and collected are always helpful. I think it allows us to understand what's happening, some sort of aspect of physiology that we can then address, particularly go then from, literally bench to bedside. So it is absolutely important that we can work on those samples, particularly obviously human samples. Nothing quite emulates it in the same way.
Yeah, if I might
Really,
just add.
sorry, go ahead, Matthias.
If I might just briefly add, absolutely. So particularly in the context, as we already discussed, of the immune system and modifying it, making these cells safe. For someone who has type 2 diabetes in the end stage, they're similar to someone who has type 1 in that their beta cells just do not produce enough, for sure, and in some cases almost none of the insulin that these patients need. So if we were to generate cells that are really tolerated well, very safe, very efficient, and actually I have in mind self-healing cells that respond to stressors and then can activate a counter response. If we get to this point, then I think at some point patients with type 2 diabetes can also benefit from this technology.
Fantastic. I've got a really interesting question for yourself, Matthias, here. So we've got someone here asking, could you use your own stem cells to avoid the rejection issue?
That's a fantastic question. The answer is maybe. And the reason for that is that if you have type 1 diabetes, the reason why you have diabetes is because the immune system has decided to destroy the beta cells for whatever reasons. We don't fully understand it yet, but it's the beta cells that are being eliminated, and the cells sitting next to them, the alpha cells, as I mentioned before, they're not being destroyed. And so if you take your own cells, yes, if you would have a normal immune system, we could get your cells and we can give them back to you, and they likely would be functioning, and the rest of the immune system would say, okay, fine, that's great, that belongs to us. The problem is these cells that have decided to destroy your beta cells, they still would destroy your beta cells because they say, hey, look at this, another beta cell, let's go and take it out. So it depends on essentially if you have an autoimmune disease versus if you have something more akin to type 2 diabetes, where your diabetes depends on other factors not related to the immune system.
So someone like me, type 1 diabetic, using my own stem cells wouldn't necessarily be helpful because my body would just see it as a target. Again,
yes. We would have to modify them in a ways that the immune system would not detect them or tolerate them. Yes.
Okay. So it's still it's not an easy solution, but a really, really good question. Any more of those, please do keep them coming in. And then I'm not sure who's best to answer this one. So whoever comes up with an answer quickest, go for it. So could the glucagon cells be transplanted at the same time? So these are the alpha cells that we've been talking about, folks. It would make sense to have them both at the same time.
So they are actually, with human islet transplantation, they are actually within the islet. So the islet actually contains 1,000 cells. 80% roughly are the beta cells. Roughly 20% or a little bit less are the glucagon producing cells, and they certainly help. There are other cells, but those are the two main classes. And yes, whoever asked that question, they're absolutely right. It does help. I think there are some studies which show that actually the glucagon response to hypoglycemia is partially restored as well, which is a major problem in somebody with type 1 diabetes for a number of years. That can often be blunted, that response.
And the same is true for stem cells. It wasn't probably clear when I described it, because we generate this progenitor cell that gives rise to beta cells, we're also producing the alpha cells in one go. It's not a different protocol, so they come along for the ride. So the islets, the stem cell-derived islets we are transplanting, do have alpha cells as well.
You get the whole gang all in one. So a really interesting question here again. So this one's probably best to you, Matthias. I've heard about beta cell regeneration too. How's that different, and where is the research at with that?
Great question. So we distinguish between stem cell therapy and regeneration as endogenous regeneration, meaning you still, and this is also true for patients with type 1 diabetes, often they still have some residual function left. We know this because there's this molecule called C-peptide, and we can sometimes find this in some of our patients at a very low level. So yes, you have lost most of your beta cells, but some of them are there. But if you are diagnosed very early, which many are, by the time that you still have significant beta cell masses, there is, at least in theory, the ability to take that mass of the beta cells that's still there and expand it. That's what we would call regeneration. These cells either could be induced to proliferate, meaning to make more of themselves. It's very hard to do for the beta cells. There's a lot of breaks built in to prevent exactly that, the expansion of those. Or one could take cells that are not yet beta cells and turn them into beta cells. The problem with that is that you have to, if you use molecules to do this, you have to just very specifically direct them to the cells where the activity needs to occur. And at the same time, even comes back to the question about can you use your own stem cells? Even if you make more beta cells, the autoimmunity will still destroy them. So all of this will only be possible in the context of modulating your immune system.
Mike asks, why are the islets inserted into the liver rather than the pancreas? This one's probably for you, Shareen.
That's such a great question. So the liver is, we're able to actually reach the liver under radiology guidance very easily. And the patient actually doesn't require an anesthetic, they're just sedated. And it's a pretty straightforward procedure. The pancreas is really difficult to get to. It also has digestive juices in it. And actually, if you tried to put the islets back in to the pancreas, if you managed to get there, which would be difficult, they would all probably be digested. So that's why the liver is used. The other thing to say really is that the liver is is probably the best place that we have at the moment for transplanting islets. When we put them under the skin, very often what happens is there's a huge reaction, something called a fibrotic reaction happens, and they wall off the islets, and they're not particularly, people are still doing a lot of work to try and improve implantation under the skin in the subcutaneous tissue.
Oh. Well, I was really excited for my carrot and coriander soup after this, but the digestive juices I'm going to have a sandwich instead. You don't usually have such a visceral reaction to words like that. Right, so are there any trials happening right now that we can get involved in? We've sort of briefly, we've touched on it in part throughout today's webinar, but what's happening right now that people can get involved with?
There's lots of trials. I think if people have severe hypoglycemia, then it would be useful to go to their local hospital and see if they might be considered for one of these trials. So across the UK, for example, there are major hospitals in London, Newcastle, Oxford, Edinburgh that are actually recruiting people for example, the Vertex trial, which is the stem cell-derived islet trial. And they may or may not be people who meet criteria, but certainly I would encourage people to go to their local hospital. But other than that, there are other trials as well, obviously. So there are trials where people might could give, for example, blood samples, take part in the biosamples where which means that people like myself and Matthias can understand the condition a lot better. And there might be other technology trials as well, using, for example, hybrid closed loop systems or other trials that people should find out about.
Fantastic. And I'm guessing, the charities, Diabetes UK, Breakthrough 21D, all these resources are sort of almost starting points, gateways for people to get connected to the different things that are happening locally. I remember, a few years ago now, maybe 2018, 2019, Diabetes UK arranged for me and a load of people to go to somewhere at Southmead Hospital in Bristol, where a lot of research was happening, which was really, really interesting, because when we just sort of like pick up our insulin from the pharmacy or get it delivered, as you can do nowadays, you don't really see what else is going on, and sort of at a consumer end, let's say, it can almost seem like a very slow progression. But if it does interest you, then you can find buckets of exciting things out there to stay really on the ball. And and if you do that, then you're more likely to be making yourself known to the people that are doing the research, so you could sort of find yourself involved in it somehow down the line anyway.
Absolutely. And I should state, that there are different trials and different studies for different people at different stages of their diabetes. So for newly diagnosed people with type 1 diabetes, there are what we call the immunotherapy studies, which Diabetes UK have really driven to set up. And many of these studies involve maybe 2-3 injections to try to preserve the beta cells within the islets. Because we know that if there is even a small functional reserve, that can really protect from the progression of complications in the longer term. So there are multiple studies which people can find out about, as you said, using the resources out there.
Just a couple more minutes left to go, so we'll quickly race through any other questions that have come in. So eventually, do you think there will be one beta cell treatment to cure type 1 for everyone, or might they work better for some than in others?
I'll give that to Matthias.
Thank you. Great question. We already touched on this. Can you use your own cells? We said for autoimmune diseases, it's going to be difficult. So what is the perfect treatment here? Is there going to be just one stem cell that will give rise to all of the treatments that we have, or one protocol? It is interesting that because we are trying to replicate what the embryo does when it forms the beta cells when we are in our mom's womb, many of the protocols are very, very similar. There are differences still, but it's not that much. My guess is that there's going to be a number of companies who have slightly different protocols that will come to the fore, and we already know that there are some out there. And there are going to be slight differences, and I think they're going to start modifying them as we go along. Again, the analogy is the car that changes over time. So I do believe you're going to have several different versions of it over time, and I think they're going to get better over time.
And then it seems to be the thing that anyone comes back to when we mention the NHS and healthcare, but is it quite a big cost implication with creating beta cells, and indeed the whole sort of procedure, the transplant, getting them into the patient, and will the NHS be able to afford it eventually?
Let me start, and I'm pretty sure Shareen is going to have something to say about that. Right now, it's expensive. You have to do this under what we call GMP facility, good manufacturing procedure facilities. You have to use molecules and everything that is, of course, safe, and a number of biologically active molecules. The serum, the media, all of this stuff is really expensive. Now, once you get to mass production, I do think that the price is going to come down. And once we see, again, that it works in many patients, I can see this becoming significantly more affordable than it is now in the very early stages when it is first rolled out. So I'm optimistic, but I would like to hear what Shareen has to say.
I agree, Matthias. I think when we develop the expertise to make stem cell islets at huge scale, that will, whilst maintaining the proportion of live cells as well, then that will make a huge difference. Also, I think it's worth saying that if we can eliminate the anti-rejection drugs, which are, again, very costly, then that might, in the longer term, actually impact favorably, again, on cost.
And the last point I'm going to say is insulin
Oh,
is also not for free, right? So if you don't have to take it anymore on a daily basis and you have to take multiple injections throughout the day, this would be a cost saving as well. So at some point, it actually might, the cost will come down and it might actually be available on large scale.
Absolutely. And from an economic perspective as well, you know, if we can reduce the progression of complications, we actually reduce hospital admissions and cost in that way as well.
Fantastic. Well, both of you, thank you so much for all that information today. That's Professor Shareen Forbes and Professor Matthias Hebrok. Thank you both for joining us today and answering the questions, being so open and sharing some really, really exciting things. So if you're watching along wherever you are in the world, this will be available to watch again. You'll be able to find out how to do that through the Diabetes UK gang, and there's some interesting links that have been posted in the chat, different clinical trials that you might be able to get involved in, and also a link to the Diabetes Grand Challenge, the Type 1 Diabetes Grand Challenge, let's get it right third time, website, where there'll be plenty of information. But we've just hit 2 o'clock, so that is the end of the webinar. So thank you again, both professors, for speaking to us today.
Thank you for hosting. That was fun.
Thank you
so much.
Jamie.
It's been absolutely amazing. Thanks, guys. So let's hand it back over to Lucy then. Thanks for the opportunity.
Yeah, I'm going to close. So thank you so much, Jamie, for doing such an amazing job with the questions. Matthias, Shareen, absolute masterclass on how to explain a really complex area so clearly. Thank you, everyone. This will be available to watch back, and we'll share how you can access it. So have a great weekend.
Thank you. Bye-bye.
Bye.
Bye.